I will show you another idea from a student. The first step is the chemical synthesis of a non-natural amino acid linked with a DNA base. Here is an example. This non-natural amino acid with COOH and NH2 groups is conjugated with a thymine. You can couple a different amino acid with a different DNA base. Since there are 4 DNA bases, 4 different amino acid conjugates can be prepared. If you need variations more than 4, you can connect 3 bases to 1 amino acid. One example is shown here, which has 3 bases. Such design allows you to prepare 64 different conjugates. The next step is the chemical synthesis of a template single-stranded DNA, for example, GACCT. When you mix this DNA with the base-linked amino acids are all aligned on the DNA strand. Depending on the sequence of the DNA, addition of the peptide bond-forming reagents would form a non-natural peptide on the DNA. By changing the sequence of the template DNA, you can control the sequence of the peptides. A similar concept is now called DNA template synthesis. Reactants A and B are each conjugated with a DNA single strand. When the red DNA is complementary to the blue DNA, A and B come close to each other, shown here. That's why the reaction between A and B is promoted. When the red DNA is not complementary to blue DNA, a DNA strand complementary to both can be added to the reaction mixture. This black DNA brings 2 molecules together and promote the reaction between A and B. For example, Professor David Liu in Harvard University and his co-workers performed this experiment. What you are looking at here is a mixture of 12 starting materials. Each of them is tagged with a particular DNA sequence. The mixture undergoes the multiple reactions in a single solution to generate exclusively a set of sequence-programmed products, shown right here. For example, blue starting materials react with each other because these 2 DNA strands are complementary to each other to form a double helix. If these molecules were not tagged with DNAs, analogous experiment would generate an uncontrolled mixture of all possible products. Such DNA template synthesis has been allowed for many years. However, Professor David Liu came up with an even better idea to do DNA template synthesis. They prepared a DNA-encoded chemical library. Let's assume that 3 DNA strands are connected with chemical X. You can make a lot more than 3 in a real experiment. Add 3 reagents tagged with 3 different DNAs. Let me point out error here. The C should be shown in brown, not in red. Anyway, these reagents react with their corresponding molecules to form these 3 products. Because yellow DNA is complementary to yellow DNA, and then green DNA is complementary to green DNA. Next round of the reactions generates these 3 products here. These 3 products. D goes here, the E goes here. You get these 3 products only. You can do more. Consequently, you can generate a library of chemicals which is programmed with DNA sequences. You can select active molecules from the mixture. So let's suppose this one is active. You can select this one. Selected molecule has a DNA strand attached to it. The PCR amplification would amplify the DNA strand of the active molecules, which then serve as starting molecules for another cycle of selection. Since the structure of the molecules are encoded by DNA sequences, the analysis of DNA tags reveals the structures of the active molecules. As a proof of the concept, they synthesized 65 synthetic macrocyclic molecules, shown here. They then selected and amplified the active molecules. The experiments led to the identification of this molecule as a selective binder of carbonic anhydrase. This technology may be useful for drug discovery. Thanks for watching.